So in today's class we start with the enhancosome complex. So the first thing, just to summarize it once more again, you have sequences known as promoter and sequences known as enhancers. So these are the promoters and enhancers. Promoter, you know, is the place where RNA polymerase and other transcription factors binds, and there is also a sequence known as enhancer. So in this enhancer, what happens is essentially you have some other transcription factors. Say if over here they have given a transcription factors, the name is LDB1. There is another transcription, general transcription factors known as GATA transcription factors. They bind to the enhancer region, and one binds to the polymerase region, and it forms a bridge. And when it forms a bridge, this complex known as enhancosome, this enhances transcription. This is how it folds. The DNA, as you can see, it folds. Enhancer comes on the top, and this type of a complex is known as a enhancosome complex. E N Han CO Some complex. My PhD thesis dealt a lot with these transcription factors and enhancosome complexes. So this type of a complex, LDB1 and LDB1, it is known as mediator complex, and all these orange boxes, so they are the mediator complex. What they do, they link the enhancer and the promoter, and this is what you find here. This is the basically the pre-initiation complex which is formed over here. The initiation complex is formed here. Then you have the other mediator complex. What is the function of a mediator complex? Now, as you know, this is the DNA which is coiled around the nucleosome, which I explained in the last class. It's coiled around the nucleosome. So this DNA is the promoter region or the enhancer region. It is not accessible to your transcription factors and RNA polymerase. So these mediator complex, they are enzymes. They're enzymes. What they do, they sort of move these things and make this DNA. As you can see, this DNA is made accessible over here. The DNA becomes accessible, and as a result of which this enhancosome complex is formed. And you can, a molecule known as cohesin, it binds these this complex. It binds this complex. So once this complex, and there are the as you have read that all these transcription factors, transcription initiation factors, they bind over here. I think this will be taught by Zarka madam or Kaushik sir. This transcription initiation complex will be formed over here. Now, if this region is bound by the nucleosome, this is not accessible. it is paused. Transcription will not start because this is bound over here. Now there are factors which modify these histone tails over here, acetylates them, and so this nucleosome is released and the block is removed and this starts to elongate. This is how transcription elongation takes place because of these expression. So depending upon in some cell types, possibly this enhanceosome complex factors are not expressed, and so this particular gene is not expressed in that particular cell type, but in another cell type this is expressed. And so if it is expressed in another cell type, what you can expect then is that there is some gene expression of that particular gene. So this is how a differential gene expression is created. Now let us look at this particular thing. Say gene A expressed in brain and limb. This is a gene A. So you have a brain specific enhancer and you have a limb specific enhancer. This is gene A. So if the binding takes place, and this is the mRNA which is expressed in both brain and limb. In the case, this is the case where gene A is expressed in both brain and as well as in limb. So you have two types of enhancer, and you have the promoter, the specific promoter also. This is the promoter. This is the transcription start site over here. So for expression which is taking place only in the brain, you have brain expressed transcription factors, which are transcription factors. They come and bind to the brain specific enhancer. So this is used, and RNA polymerase do, and the mRNA is then expressed in brain. This limb specific enhancer is not used here. But when limb specific transcription factors are there, the brain specific enhancer is not used and only the limb specific enhancer is used, and so you have mRNA which is expressed in limbs. Such a thing is, for example, you can see it in this particular gene, the PAX6 protein. The PAX6 protein is critical in the development of several widely different tissues. Enhancer direct PAX6 gene expression differentially in the pancreas, the lens and cornea of the eyes, and the neural tube. So what they did was they attached behind the promoter. After the promoter, they removed the gene and they attached a beta galactosidase. So when you give X-gal, a blue color is stained. If the gene is expressed, if it is not expressed, then it is not. There is no beta galactosidase activity shown. What you do is basically after the enhancer, after the promoter, you remove the specific gene and you attach a beta galactosidase enzyme gene. And once when you attach the beta galactosidase gene, if the gene is expressed, instead of the gene being expressed, if the promoter is stimulated, the gene is not expressed. What is expressed is beta galactosidase, and then when you add X-cal, you see a blue coloration. So wherever you see a blue coloration, you are sure that that particular gene is expressed over there. That is its specific. So you have a pancreas enhancer over here. Then you have a lens and cornea enhancer, neural tube enhancer. So depending upon which tissue and which transcription factor binds, you have expression either in lens or expression in pancreas or expression in neural tubes. That also directs which type of transcript is also formed. So this is known as modularity. So this is a pancreas module, this is the lens and cornea module, this is the neural tube module. So enhancers have got a modular design. Then another thing is combinatorial association. That is many people call it as a synergistic effect. So expression of a gene is a result of, say for example, this PAC6, this particular somatostatin gene in the pancreas. You have PDX1, PBX1, PDX1, PBX1, PDX1, PAX6, CREB, all these together along with TVP, they all of them together enhance the transcription. There is another effect known as synergistic effect. That is, say a transcription factor A gives activity X. A transcription factor B alone gives transcription activity, say X by 2, half of that. But when you add, say for example, A plus B, if the effect is additive, then the transcription becomes, the enhanced expression level becomes 1.5x. It becomes 1.5x. 3 by 2x it becomes. But this is additive effect. But when it is synergistic effect, A plus x, it perhaps becomes 100x. Alone, they are unable to give an expression high. But when you add both of them together, you find them in immunoglobulins. A lot of all these things happening in immunoglobulins. You get a synergistic effect. A huge amount of expression starts to take place. Then you have, like enhancers, you have silencers. If the silencer, instead of enhancer, there is a silencer. If a silencer comes, then whatever expression of the gene is there, it stops. You can read this mouse embryo containing a transfer of L1 promoter and a portion of the neuron specific L1 gene and LAG-Z fused. So LAG-Z and L1 promoter. No NRSE sequence. You see, the blue color shows that this galactosidase or the LAG-Z is expressed throughout the mouse. But when you have an NRSE silencer sequence somewhere attached, this actually blocks the expression of this promoter, and there is no beta galactosidase expression. So there are genes known as silencers also present here. So all these silencers, enhancers, they have got cis regulate. Cis means pashapashi. They are on side by side. So this is a 6 configuration. And trans means one strand here. This is a trans configuration. So cis regulatory elements means they are side by side. So therefore these are all cis regulators. So I end my class today over here. Do you people have got any questions? Please do ask my questions if you have any questions. I have to go slow because these are a lot of concepts to grasp. One day I don't want to overload you people. So try to read these things very carefully, meticulously. Since we have got lots of some time left, I've got less amount of things to do, I can do these things slowly. I can talk with these things slowly. So what I will try to do is I'll try to show you a video of how transcription takes place.
Good grammar and spelling are important, but if you want to write essays that inspire,
our cell is like a factory. Now inside the factory there are several machines producing its product, and this product is going to the market, and market has a certain demand according to which these machines are producing its product. Now inside of the cell also there are necessity of proteins in order to sustain the developmental, the growth, and also the physiological processes of a cell. So that's why all the time DNA is going to RNA, and from RNA protein is made. Now in this process from DNA to RNA to protein, the need is variable. Sometimes a cell require one protein more than the other. So all these processes of DNA to RNA or RNA to protein could be regulated as per demand. And that's why regulation of transcription and translation processes are so important to understand. In this video we would talk about transcriptional regulation. Just like a fan has its own regulator, and turning the regulator we can change the fan speed, a transcriptional machinery, that means from DNA to producing mRNA, also has regulatory mechanisms. Some regulatory mechanisms are trans-regulatory mechanisms, which are protein-based or based on transcription factor binding, and some are mediated by cis-regulatory elements, which are also present in the DNA nearby the gene or far away from the gene. And I'm going to talk about those cis-regulatory elements in this video and how they regulate transcription. Now here is a typical simplified diagram of a gene where yellow is the gene body, and now the first regulatory element to talk about is a promoter. Now promoter is a short regulatory sequence just near to the gene body, either at this and where the RNA polymerase 2 bind and transcription initiation happen. For transcription to initiate, at least a minimal promoter should be there. Apart from promoter, there are other sequences known as enhancers. Enhancers could be both sides of the gene, either upstream or downstream, but they could be like as far away as in order of 3-4 kilobase pairs, and still they can regulate the gene expression from a particular gene promoter and from the gene body. So how does enhancers work? Actually, the general transcription factors of TF2D family binds to the promoter, the minimal promoter, and recruits Pol2 in that promoter. Now upstream enhancer elements bind to specialized transcription factor. Specialized transcription factor can interact with cofactors. Now these cofactors could be either co-repressor, or some cases these factors could be coactivators. If it's a coactivator, these upstream enhancer sequence undergoes a DNA folding mechanism such that these cofactors and specialized transcription factor can interact with this Pol2 complex and thereby regulate the frequency of translation, or how frequent or what is the speed of the transcription would happen. Now inside of the cell, the DNA is not free. So DNA is in form of a nucleosome arrangement. So here I'm showing a gene X and two enhancer of it, gene A and gene B. Even though gene B is pretty far away from this particular gene X, enhancer A and enhancer B, both of them has certain amount of influence on the expression of gene X. Now I need to show you guys the different chromatin states of an enhancer. Enhancer is active when the enhancer is accessible to specialized transcription factors. That means the nucleosome is somehow translocated away from that enhancer region. Now it could be also primed enhancer, where the enhancer is accessible but not the transcription factors, the specialized transcription factors are just not bound. It could be also poised, that could be opened or closed, so bidirectional markers are there in this state, but exactly they are not accessible to the transcription factor right now, but they could be modified in near future depending upon the need of the cell. Now, different enhancers could influence the gene in a differential manner depending upon which tissue they are present at. For example, inside a neuron, let's say there is an expression of gene X, and gene X is very important for survival of the neuron. So gene X is modulated by the enhancer A here, but not by enhancer B. But let's say we consider another cell, intestinal cell, and here we see a different scenario. Enhancer A and enhancer B both regulate the expression from gene X. So depending upon the tissue, the enhancers that regulate the transcription could be also different. And that leads to the heterogeneity of gene expression in different tissues. So have you ever wondered that how to identify this enhancer sequence? Now, if you know a certain transcription factor that binds to this enhancer region, then definitely you can pull down that with an antibody and then sequence the region where it binds. That would give you the enhancer sequence. Otherwise, there are many other approaches, bioinformatic approaches, histone modification seeks, chromosome conformation capture based approaches, and DNA-seq. All these things are there to find the enhancer sequences. Now, how to understand that one enhancer is active and one enhancer is inactive or closed state? So in an open state, there are specific marks on the enhancer that tells us that it's in an open state, which is very different from the marks present in a closed state. So if you even look at the different histone modifications in a chromatin region, which is a putative enhancer, then we can understand, okay, the enhancer is there. In my other videos, I would be talking about
So this is basically you don't need more than this. This is this part, and then there is also another video which I just now saw, which I found very interesting to show. Just to keep yourself involved, nice looking things. Just
by controlling which genes are active, a cell can take on special characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA-protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
So this was basically my class today. Let me just check the number of people here. So we have 13 people in the class. 2 or 3 of them have left. Anyway, this is the number I write down. And okay. Thank you. Anybody has got any questions? কারুর কোনো কোয়েশ্চেন আছে?
না স্যার।
ঠিক আছে, তাহলে আজকে আমি শেষ করছি। ভালো থেকো সবাই।